Hello Bio20 and welcome to your refresher lesson on enzymes. Today we're going to talk about the general purpose of an enzyme again and how it'll aid digestion. So the idea is that you're going to define an enzyme based on the previous knowledge you have and the video you watched with the Amoeba sisters, as well as you're going to continue to look at what happens to enzymes when they're not working in their optimal temperatures and pHs, defining the word denatured again, and again talking about how enzymes are going to play a role in lowering the activation energy so we can digest our food. So as a reminder, what is an enzyme? An enzyme is a catalyst. So again, a catalyst's job is to increase the rate of reaction. They can be reused over and over again because they do not change because of the result of the reaction. Now, a catalyst is the general term that's used. In biology, we go specific and call it an enzyme. So in the picture there, you can see an enzyme with an activation site and a substrate. You will remember that an enzyme, is specific to the substrate. So it's essentially fits together, kind of like puzzle pieces fit together. So an enzyme is an organic catalyst. So organic meaning it contains carbon that's found in the human body. We have enzymes in our body that break down carbohydrates, so big sugars, so that it can go into the mitochondria as glucose to do cellular respiration. Enzymes are made up of proteins, and their role is to help our body create those chemical reactions to break down our food at body temperature. If we did not have enzymes, every time we wanted to digest our food, one of the ways that we can break things down or hurry up a process is by raising the temperature. You may remember that from grade 8 or grade 9 science where you looked at the rate of reaction. All right, so the rate of reactions means you can stir it, you can make it into smaller pieces, you can add heat. So if we were to have a reaction in our body every time we ate and we didn't have enzymes, we would have to make sure that the pieces were very small, that we could somehow turn them up, and then we would have to raise the temperature in our body in order for those reactions to speed up. Now that's really bad. You do not want to raise the temperature of your body. Your body is the temperature it is for a reason. So that's where enzymes come into play and help us break down food in our body. So they're necessary because chemical reactions on their own are very, very slow. So again, I just spoke about this. One way to increase the rate of a chemical reaction is to increase temperature, but that's bad. It damages our cells. So when you raise the temperature in your body, you actually do something called denaturing of proteins. Now we learned last lesson that proteins come in a bunch of different shapes. One of them is structural and one of them is globular. They're made out of lots of amino acids folded up. When we denature a protein, it means that we unfold that protein. And that's bad because sometimes those proteins can't fold back up into the way they were supposed to, and they can no longer function properly. So we do not want to raise the temperature in our body because then we would possibly denature these proteins. So we use enzymes to speed up the rate of our reaction without increasing our temperature. As we've talked about before, enzymes are substrate specific. So substrate is whatever it's trying to break down or build, because enzymes can build too. But in the world of digestion, we're going to only talk about them breaking things down. So the substrate might be something like maltose, where we're going to break maltose into something smaller. What ends up happening is that substrate attaches itself on something called the active site of the enzyme. The active site is the area that matches the shape of the substrate. It's a 3D structure that specifically matches what the substrate structure looks like. We call this the lock and key mechanism. Now, another thing that's really important as we move through this chapter that you need to understand is the unique naming of enzymes. So enzymes are given a very unique name based on the substrates that they act upon. So if I have maltose, M-A-L-T-O-E-S, I'm going to have an enzyme that uses the name maltose, but instead of calling it maltose enzyme, I'm going to change the end of the name to ASE, and I'm going to call it maltase. Same with sucrose. You can see sucrose plus water. We're going to break apart sucrose. We're going to do hydrolysis using water to cut something. So we're cutting sucrose into fructose and glucose. In order to do that, we want to speed up the rate of reaction. So we use the enzyme called sucrase. Sucrose is the substrate, sucrase is the enzyme. Now in the world of enzymes, they constantly would break down things if they could, or build things if they could. So we have to sometimes what we do called inhibition, meaning stopping them from doing things, okay? Inhibition or inhibit to stop. So there's two ways that enzymes can be blocked. The first one is called competitive inhibition, okay? Competitive inhibition means when something that is not the substrate binds to the active site. So something comes in, binds to the active site, and now that substrate no longer fits in the active site. Non-competitive, on the other hand, is when something binds onto the enzyme somewhere, but then changes its 3D shape. So now the enzyme no longer is the shape it was, so the substrate cannot fit on it. So that's the difference between the two. Come up with a similarity. Competitive, non-competitive, what are they both about? What makes them similar? Think about that for a second and then write it in your notes. So let's look at competitive and non-competitive just in picture to kind of show you what I mean. So on the left-hand side of this picture, you're going to see competitive inhibitor. So there you can see something bound or was bound inside the active site, something that is not the substrate. So as the substrate comes in, it bounces off because it can't actually bind in there. Non-competitive on the right-hand side, however, goes in and changes the shape. So you can see that that yellow piece went in and was binding in up here. And what it did is it pushed on this edge and pushed it up. So this no longer fits because this groove is too deep. So this is non-competitive and this is competitive. The reason why we, again, need these things is because we don't always need reactions to be occurring in our body. So this is one of the ways we stop enzymes from functioning. Now there are problems with enzymes because enzymes are made out of proteins. So I've already spoke to you about what happens if we raise the temperature of our body. It's a problem because we denature that protein. Temperature and pH within our body is so specific, and enzymes work within specific areas because they like those conditions. So when you have an enzyme that is in a condition it no longer favors, so it's too hot, it's too cold, the pH is too high, the pH is too low, that enzyme can no longer function, and we have that unraveling called denaturing. So you can look here, the rate of reaction. This is an enzyme's rate of reaction. So here at 0 temperatures, at about 37 degrees, we see the rate of reaction take off. That's the optimal temperature for this enzyme to work at. On the other side here, we see enzymes that function at different pHs. Pepsin, an enzyme you're going to learn a lot about in the next little while, really likes working at a pH of 2. So it's a very acidic pH that's found in your stomach. So pepsin is in your stomach. It only works in a pH of 2. Trypsin, on the other hand, another type of enzyme, only really likes to work at a pH of 8. So if you put trypsin in a pH of 2, it wouldn't function. Both of these enzymes like to break down proteins. So they're in different parts of your body based off of their pHs. And if you switched them, they wouldn't function and you wouldn't be able to break down proteins. So you've already seen this video, so I'm not going to play it, but if you haven't watched it from yesterday, please go back. It's a great review. The notes that you've been taking, please make sure you're done them and make sure you answer the questions on enzymes and have a conversation about them. If you have any questions, please let me know.